All right, awesome. Thank you, Amber. So yeah, we can go ahead and jump right in. Let's head to the next slide. So before jumping into some of the picture-specific updates, I did want to give a quick update on some of the data available in Biodata Catalyst. So as you all know, we prioritize the ingestion of many high-priority heart, lung, blood, and sleep-related data sets. And so, this is just a quick and dirty summary of some of the high-priority phenotypes that we have available, broken down by number of data sets. So you can see we've got a good chunk of cardiovascular-related data sets, heart-related data sets, sickle cell disease, so on and so forth. So just to give you a brief overview of, what sort of data we have. Next slide. So this slide is more for the magnitude of the data that we have, or the size of the data. So as you can see, we have over 3.4 petabytes of data available in BDC and over 280,000 participants, over 490,000 data files, and over 150,000 whole genomes sequenced. And we're ingesting new data sets every day. So hopefully, we're excited about these numbers to go up over the next year. And I also wanted to mention that a good portion of the data we have available is from the TOPMed, or Transomics for Precision Medicine initiative. And you can get more of a feel for the phenotype breakdown and the ancestry and ethnicity breakdown of these data sets with these pie charts here. Next slide. All right, so this is a list of all of the studies by the abbreviation or code that we have available in Biodata Catalyst. And you can see here, of these that are listed, the bolded ones are actually part of the TOPMed initiative. So you can see that a lot of these studies are related to the TOPMed initiative or program. Next slide. We've also ingested several COVID-19 related data sets. So you can see those bolded here. Those are Active 4A, Active 4B, C3PO, ORCID, and Red Coral. Next slide. Additionally, we've been prioritizing the ingestion of sickle cell disease related data sets. So you can see those bolded here. Among these are the Walk Fast, Stop2, I believe Baby Hug is actually also on here. I may have missed that, as well as CSSCD. Next slide. And we also have publicly available data sets on Biodata Catalyst. So these include several data sets from the BioLink repository, so the Framingham Camp and Digitalis. We also have 1,000 genomes and a synthetic tutorial data set available. Next slide. And as always, you can check which data sets you're requested or authorized to access by going to our website here and clicking the View Available Studies. You will be prompted to log in with your ERA Commons, and that will help us tell you, which data sets you have an active data access request for. Next slide. So in order to get access to the data, so to actually be able to access the participant level data and to really start on your analyses, you will need to have an active data access request in dbGaP. And so the components of a data access request include a research use statement, a non-technical summary, and we also have Biodata Catalyst specific cloud use statement with template language available to help you compose a data access request or submit one. We also have resources available for helping to submit this, so you can always contact our help desk with any questions that you have. And we also have just added some documentation to the Biodata Catalyst Git book, which is linked here on this slide. Next slide. All right, so that was a quick rundown of, an overview of the data that we have in Biodata Catalyst. And now I'm going to jump into some of the picture-specific updates. So as some of you, I'm sure, are aware, Picture actually released a new version, which pretty much changed nearly every aspect of our platform in September of this year. And so this includes a simpler underlying data structure, allowing for filtering of search results by variable and study tags, a new genomic filtering modal, an add variables to export without filtering, a simpler select and package data process, visualizing single variable distributions, and of course, all of these new new features are available through Picture's open API. So I'll jump into some of these just to highlight some new updates here now. Next slide. All right, so one aspect of this updated release is that you can now filter with tags. And so we have two types of tags. There are study tags and variable tags. And so you can see here, I've searched for triglycerides, and I can narrow down these search results by adding these tags. So I'm filtering to only results from the Framingham Heart Study, or FHS, and I'm also including the exam tag. So this helps me to locate, in this example specifically, longitudinal variables that are available. So using the exam, so you can see then in the results highlighted in the box there, there's triglycerides measurements for several different exams from 1 to, it looks like 28 on here. So helps to, narrow down and really get specific with your search. Next slide. Additionally, we've added a genomic filtering button. So in Picture Authorized Access, you're able to click on the genomic filtering button that is next to the search button. And this really streamlines it's kind of a one-stop shop for all genomic filters that are available on Picture. You used to have to know exactly which variables to search for, but now when you click on the genomic filtering button, next slide, please, you are taken to this modal that displays all of the fields that you can filter by. So you can see you can select a gene, variant consequence, severity, variant class, and also variant frequency. So it really streamlines that genomic filtering process. Next slide. Additionally, you can add variables to export now. So you can see next to the filter button in that actions column, there is an option to add the variable to export. So this is new because in the older version of Picture, you actually had to filter on the variable in order to have it be exported in the data frame. But now if you're just interested in viewing what data is associated with that variable, you can simply add it to your export and it is included as an extra column in your data export. Next slide. We also have streamlined the select and package data process. So instead of, if anyone remembers, the data tree that we had displayed, now we have this really nice, easy to read select and package data modal. So it provides a quick summary. So the number of participants that you've selected, the number of variables, and therefore the estimated number of data points. So you can get an idea of how big the data you're exporting is. You can also select and deselect variables. So if I'm reviewing this export, I can say, oh, maybe I'm not interested in father asthma status, so I'll uncheck that variable from the export. Next slide, please. And something that we're really excited about with this that was actually released, I believe, last week, are our new export to Terra and export to 7 Bridges buttons. And so when you click on package data, it will package up your data and display a query ID, but it will also have these 2 buttons as options. And when you click on either one of these, whichever is your preferred analysis workspace, you will be directed to this new modal that gives you step-by-step instructions and provides the links to get you directly to where you need to be in Terra or in 7 Bridges in order to complete a picture export. So we're really excited to take this next step to improve interoperability throughout the ecosystem. Next slide. An additional new feature is the statistics and visualization feature. So this is called the variable distributions tool in our tool suite on authorized access. And this really allows for you to get an idea of, what the breakdown of participants is or what the distribution of participant measurements is based on the cohort you've selected. So here in the screenshot, you can see that I can get an idea that, the majority of the participants in this cohort do not have COPD. So depending on my research question, perhaps I need to adjust my cohort that I've created. Next slide. All right, and as I've mentioned before as well, even though all of the things I talked about were updates in the UI, we have also created updates for our Open Application Programming Interface, or API. And so this allows users to query Picture using code. And we actually have publicly available code on both Terra and 7 Bridges. And you can use Python or R and Jupyter Notebooks or RStudio, whichever platform or interface is your preferred method of development. And of course, our code is under an Apache 2.0 license, so everything's open source. And, you can also see our, publicly maintained GitHub repository, or publicly available, sorry, actively maintained GitHub repository linked here on this slide. Next slide. All right, so that concludes my, quick introduction on all things new with Picture, all things new with data and Biodata Catalyst. I'd be happy to answer any questions, Amber, either now or later, whichever works.
Yeah, thank you, Emily. If anybody has questions, we can open it up and take them now. Okay, doesn't sound like any questions right now, but thank you, Emily. A lot of cool new features, and we look forward to the year ahead. We'll move on, and our first guest researcher, Jean, is going to speak. I think he's on the call. Yeah, there he is.
I am here.
Cool. Okay.
Hi. Thanks for having me. I'm part of I was part of the first cohort of fellow, and I'll talk a bit more about my project there. If you could go to the next slide. So my BDC project was part of a bigger effort to improve and showcase pangenomic, which is in our case the use of a better reference genome. So it's where we integrate right from the start information about other genomes, hence the term pangenome. So in my case, I made a pangenome that contains some of the most challenging genomic variants, the structural variants, the bigger and more complex ones. And I used that pangenome to genotype them from short-read sequencing data. And so the message of this paper that was published almost exactly a year ago was that we now have a pangenomic approach that is not only more accurate, we knew about this, but also it's as fast and efficient as traditional methods. And so to show that, I used the BDC ecosystem to analyze thousands of samples with the whole genome sequencing data. So it was on one hand, a proof of concept of the scalability of our tools. And at the same time, we've produced a unique SV structural variant catalog across the diverse cohorts we had access to through BDC. So that's kind of the research highlight. And on the next slide, I think I describe a little bit more about, if you can go to the next slide. I describe a bit like what I used in the BDC ecosystem. So I had implemented and deposited my workflows in DocStore. I used data that I imported from Gen3. That's the second component, and like using the big data set, or at least part of the big data set that Emily mentioned, the TopMed data set, I used a subset of the MESA cohorts, the diverse cohort that I analyzed, and I did all the computation and the analysis on Terra. And so I think the advice I would give to someone who would like to do something similar would be first to start small, to really test the workflow and what you're about to run, test it on a really small data set first, testing it locally, starting with just one sample if you're going to scale to a lot of samples. So that actually took a lot more time than the actual analysis of the thousands of genomes. I think on Terra, because it's so elastic with the cloud, it took maybe a week or two. But the first part of making sure everything is going to not be too expensive, it's going to work and can scale is very important and takes more time. So I would say, I would recommend really spend a lot of effort on this step. The second advice, again, from experience, maybe something we realized after the fact, is to try to keep things organized from the start as much as possible. So the components at least I used, especially DocStores and Terra that I use the most, and I'm sure Seven Bridges also has this, have features that can help you organize data in table and keeping track of what you've run, where the output is, what the results are, but also tag your workflows to keeping track of what you've run and which version of each tool and which anything. So all of this is available. So I really think that should be used to really keep track and document everything. And also the writing analysis in notebooks can also help you and everyone else who wants to look at what you've done, understand what you've done. So I think that's also a very important one. And the last advice, I think, is a no-brainer, but to keep an eye on the costs. But I just wanted to say that sometimes some costs may be less obvious than others. So I think it's maybe good to be a bit paranoid and always check, oh, have I left something running and it's costing money, or how much is the storage costing me? That's something I've also kind of realized at some point in my experience, that some big files, temporary files are still there and costing money. So I think that's the last thing, but yeah, to think of what could be costing money and look at the guest reports and other costs. And I'll be happy to answer any question now or at the end about the other research or my experience with BDC, which was a great enabler for me, at least for my research. Thanks.
Thank you, Jean. Again, we open the floor for questions if people have any. Otherwise, again, we can take questions at the end, but we'll take some time now. Okay, and not hearing any, we will move on. Before we start the Gen 3 updates, I am going to launch.
Hi. Sorry.
Oh, yeah, go ahead.
Hi, this is Kritika. Hi, I've been working with Biodata Catalyst, and I don't know if this is the right platform to ours, but I wanted access to CRAM files for a particular study. And I was wondering if anyone could help me like gain the right access, or where can I did see that there are a couple of apps for SRA toolkits, but as far as I understood those apps, maybe my understanding is wrong, but as far as I understood those apps, those are basically for converting an existing CRAM file to FASTQ files and pre-processing them or post-processing them and stuff like that. But if I want to download CRAM files, or if they're already available on Biodata Catalyst, where can I locate them? Thank you.
I would say that's a question for one of our platform reps on the call. Not sure who might have the best answer, but
I can jump in. It depends on what analysis platform you want to send the CRAMs to. So for instance, if you are using 7Bridges, I'm a rep for 7Bridges, you could use the data browser feature to find NHLBI hosted CRAM files. But if they're
These are TopMed files. These are TopMed CRAM files.
Oh, so that's absolutely
TopMed,
that's absolutely the way to go. So I'll message you my email, and then we can sort of touch base about how to do that.
Perfect. That would be great. Thank you so much, David.
No problem.
thanks, Dave. Any other questions? Of course, it can be related to the picture updates or Jean's research, but if you have questions in general, this is also a good time to bring them up. Okay. So before we jump into Gen 3's updates, which we will do in just a minute, I'm going to launch a poll. If I can, there we go. Always a little tricky. So I'm going to launch a poll for something that we are calling a data-a-thon. So if you've heard of code-a-thons or hackathons, very similar. And we've heard some interest in perhaps having a data-thon for BDC. And if you think you might have interest in this, we're just trying to gauge the interest so we can maybe plan something for next year. And if you aren't interested, we'd like to know if you might want to be a team leader or help organize. Of course, you can definitely say no to that. But we do have some people that have expressed interest in getting hands-on and leading something like this. So I'll leave the poll up while Gen 3 provides their update. So you have some time to read the questions and answer. Dave, do you have anything to add to the data-a-thon?
No, I think you explained it pretty well. Please indicate if you are interested in being a team leader. This is something we want to get done in 2023. So we're still in the early stages of planning it, but we would like to showcase the data and as well as the tools that we have on the platform. So if you're interested, and also don't shy away if you're not a coder, we're going to make it accessible to a variety of backgrounds. Thanks.
Thanks, Dave. And again, this in no way obligates you into participating. If you think you might be interested in just learning more about what a team leader does, we can talk about that. And if you find out you're not interested, that's more than fine. So no obligation here. Again, just gauging interest. So now we will get into Gen 3's updates. Justin's on the call to provide those for us.
Hey, thanks, Amber. So yeah, as Amber said, I'll be walking through a high-level overview of the Gen 3 ingestion, more in its current state, but I'll also be talking a little bit about some of the future enhancements and a couple of important projects that have been implemented within the past year. Next slide, please. So Emily already gave a great overview of the data available in BDC, but this slide gives a little bit more context. It gives a bit more of a quick summary of the subject, study, and the file breakdown by our top 5 sources. So we have Freeze8, WHI, the CHD, which is our PCGC and SRA data, and then the dark blue comprises the other remaining data sets. And so in Gen 3's current state, there are about 162 studies. The top med, parent, and whole genome sequencing data sets are some of our main ones. We also have BioLink. Again, I mentioned PCGC, and quite a number of COVID-19 data sets. Of course, many others as well. There are 4 main exchange areas from top med. Those are the freezes 1, 8, 9, and I believe 10A. And so at this point, Gen 3 has really indexed over about 3 petabytes of data across over about 500,000 files. And over the past year, we've experienced about a 10% growth in the project or study ingestions. And then we've had about a 4% growth in the total number of subjects, but we've actually had a 17% growth in the number of files. And that growth is primarily attributed to one of our PCGC studies. Next slide, please. So we've made multiple improvements to the data ingestion pipeline. So, we consistently have had shorter time periods between releases to production. The QA automation pipeline has had a number of different improvements to include more QA checks. And the ingestion pipeline in general for dbCAP studies has been improved to pull data directly from an eUtilities function. More on that shortly, but and then, some of the upcoming changes, we've had plenty in the works, but just a few that we're listing here. So we're looking again to make more automation and streamline the metadata or structured data pulled from dbGaP and other sources. We're also looking to implement possible imaging functionality and then potentially have crosswalks between different platforms and commons. And then, of course, interfacing with FHIR data. And then the chart on the right is just a graphical representation of the file count and the size in petabytes of the amount of data that's available in Gen 3 over each of the different releases. Next slide, please. And so one of the tools that was really worked on this year is a metadata extraction tool. So there is metadata extraction from dbGaP that is currently being integrated with the eUtilities extraction tool. And this extraction tool was created by dbGaP, and it's being used to handle multiple different input formats. And really the long-term vision of this is, again, to have this automated. And multiple platforms have been working with dbGaP support to send over our different requirements lists for ingestion. And the end state, what we're looking for this is to have an input of all of our accession numbers. These would be like our PHS numbers, et cetera, and then output each of the different files for submission. And then we could hopefully have that more publicly available. And then in our current state, eUtils scripting into Gen 3 is really privatized for the Gen 3 support team. But again, we're looking to expand that and make that more publicized. And then, of course, just a quick example of how the script might look as we're running it at the bottom of the screen there. Next slide, please. One of our biggest improvements we've done is tweaking and modifying the discovery page to make it a little bit more user experience friendly. So a lot of our changes were UX changes, including we added a few new columns. There's better formatting. There's different tabs in the sidebar. I believe we also added that the search bar is more central on the page, stuff like that. And so, for unauthenticated discovery of available data sets before requesting access, we do now have a preview option, which is really nice. So anyone who's looking for specific study data, they can go in there and they can preview what might be available. And then now this really is going to serve as our source of truth for both public and open data set level data for all ingested data sets in BDC. So it's kind of our new one-stop shop for looking at public data sets. And that's pretty much it for the Gen 3 side. Just a couple of things we touched on here. There are definitely a number of others, but so if you have any questions or interested in learning more, I could answer some questions now, or we could again touch base at the end of the presentations.
Thanks, Justin. Again, we'll open it up for questions if anybody has one. Okay, we will move right along then. Thank you again, Justin. Jasmine is our next researcher.
Hi, everyone. I'm just going to talk to you a little bit today about my I was in the Fellows Cohort 3 group and what I did while I was working both on Biodata Catalyst platform powered by 7 Bridges and for my thesis, actually. So my goal was to develop a sensitive detection methodology for Plasmodium, the parasite that causes malaria in sub-Saharan Africa. Specifically, this was a TopMed project called Africa 6K, which will be digested by the platforms probably in the future next freeze. So next slide, please. Back up. I think there's a click that happened there. Okay, yeah, so the first 2 points, just to give you a little bit of context about my research, because I think it's really important for us to realize why we're doing things. So malaria is an infectious disease that causes 247 million cases and over 600,000 deaths in 2021 and the years prior. And the reason that this is important is because we've been trying to fight to get rid of it across the globe. And right now, the majority of this disease burden is caused by Africa. There's 2 reasons why I highlighted this. As you can see in this map, right next to the blurbs of information, that's where all of my research was from sub-Saharan Africa. So it's important to pick a disease that's relevant to these people and the places where we're doing the research. So my goal was to develop a methodology using only human whole genome sequencing data, obviously through TopMed, to be able to detect this parasite in these populations to retrospectively diagnose them. And as you can see through the pie charts, we actually found a really exciting thing where we could do it on the species specific level, which is really important in malaria. And we found a pretty big diversity of species across the different countries that were involved in the population. And then finally, if you want to do the last click, so we can bring the last blurb up. Obviously, it's really exciting that I made this methodology using only the unmapped reads from human WGS data. But what we found, does it actually work within the context of historical data? Because when you're building something like this, you really want to make sure that it makes logical sense. And I'm happy to report the most exciting thing about this project is the estimations we're finding are really falling in line with 2 very prevalent databases on malaria prevalence, which is the Malaria Atlas Project, which works primarily in children, so they tend to have a really high measure of prevalence, and then the WHO, which is just countrywide. And as you can see in the column right there, most of our numbers are falling directly in between these 2, which just means that what we're finding really makes sense, and we're really confident in the pipeline that I built. So moving forward, next slide. This is all well and great. I think my research is really interesting, and if you would like to read it, the next slide has the paper that is correlated with this research. But what I did may not be as applicable to what you want to do, so I want to highlight some of the things that the platform really provided for me that you could perhaps adapt for yourself. This actually already came up in conversation just a little bit ago. How I accessed the CRAM files to be able to do this research was a really big plus for me. So the easy access was when you get your sequencing data from the Broad, they just put it into a Google bucket, and we were able to completely cut out the time and cost of downloading all of our CRAM files individually by just connecting it directly to the platform, which was amazing. The next thing is there was a high parallelization that took some of the steps of my research that were taking days and in some cases weeks, and took them and reduced the time that it took significantly. So I have one example where something that took nearly 5 days on a local machine was less than 6 hours on the platform. And that's pretty useful for any type of research, not just mine. And then finally, this population from TopMed is actually shared between two specific laboratories, one at Case where I'm at, and then the University of Pennsylvania. And sharing the results and the workflows that I used was really easy to just have the collaborators log on and be able to see everything that I did. And it's important because some of the files that I process is going to be translated into other research for them. So one more slide. And so if any of those things apply to you and you're really excited about getting onto the ecosystem, here's some advice for you. Some of these things have already been said, but I think it's just really important for these kind of things to be repeated so that it sticks. The number one thing is utilize the help and resources that they have built for you. So for example, I worked on 7 Bridges. There are tons of things that you can turn to if you're feeling stuck or not getting where you want to be. There's written resources, and then obviously all of the staff have been incredibly helpful in my research. And I believe that they're there for everyone who is using the platform. And that's just really helpful to keep going. And then finally, evaluate the apps available on the public repository before assuming you need to build one. There's a massive information when you go onto the cloud platform, but always check to see if the analysis you want to do are already up instead of assuming that you have to do it yourself. And then finally, I think this was said before, create full workflows incrementally. So doing little digestible pieces make it a lot easier to determine what may or may not be going wrong instead of making like a 14-step pipeline all in one step. If you want to know a little bit more about my research or see exactly what I did on the cloud, I do have a GitHub that goes step by step through how I did my research on 7 Bridges. And then finally, this paper is currently in the final steps of being reviewed by collaborators, and we plan to submit it by the end of the year, which was my biggest and most exciting accomplishment using the cloud platform so far. And with that, I'll take any questions.
Thanks, Jasmine. Congratulations on the paper and getting it so far. I know that's an amazing feeling to just start wrapping things up and reach your goal. So congratulations on that.
I'm super excited.
Yeah.
Yes, thank you so much.
I know you.
Go ahead.
I know you've been working really hard on it, so it's really great to hear that. I like hearing the successes from our researchers.
Yeah, definitely a sense of relief. And I defend in a month. So that's the next thing that will be a great thing that comes out of my research.
Awesome. We'll have to have you back on a future showcase to talk about it.
Cool.
So any questions for Jasmine? Okay, and again, these links and a lot of the things that we've been talking about are in the slides, which unfortunately I couldn't upload through Zoom, but if you go onto the link I put in the chat, you can download the slides to access, for example, Jasmine's GitHub, the link here if you want to read more about her research. Thank you, Jasmine. We will move on to DocStore then with Ben.
Hey, everybody. Next slide, please. Okay, so brief overview or an update on DocStore. We currently have over 1,600 WDL workflows and 400 CWL workflows and tools. I should be qualifying it. That is both workflows and tools in DocStore. There is just to call attention to it, there is the Biodata Catalyst organization there so that people who have submitted as part of Biodata Catalyst can add their workflows to this organization and assign them to have them assigned to the appropriate organization. So that should be one of your first stops. Next slide, please. Wow. We currently have 53 organizations registered with DocStore that are sharing workflows. Now, I do want to highlight that researchers don't have to be part of a organization to submit workflows. So this is not a full representation. And I do need to be upfront. There's probably 1 or 2 organizations that are missing from this cloud because I haven't updated it. We've had a few new organizations added. And anybody anywhere can access and run any of the published workflows in DocStore. We also have the ability to support private workflows that are private versions of workflows so that you don't have to publish and share workflows that are under development. but anybody worldwide can access the workflows within DocStore. Next slide, please. And what does DocStore do for everybody? So it's a registry, so you have a one-stop shop to go look for potential workflows to help you with your research, either to help you that you can use them out of the box or potentially take them as a base and then work on developing them. DocStore is based on the GA4GH standards, TERS and WEST. Currently, there's integration with a number of execution or analysis platforms, as you can see on the column to the right. That includes Biodata Catalyst, both the Terra and 7 Bridges platforms, as well as another number of them. We also support local compute, be it your desktop or the local compute cluster or regional compute cluster. Those are supported. And the current workflows supported or languages supported are CWL, WDL, Galaxy, and Nextflow. And the source repositories can be GitHub, DocStore itself, GitLab, or Bitbucket, Atlassian Bitbucket. And for containers, Docker Hub, Quay.io, GitLab, and limited support for Amazon ECR. One of the cool things that I don't think a lot of people realize is that we also have integration with Zenodo and Orchid. If you are registered, if you link your Orchid ID with DocStore, if you publish workflows, those will automatically show up under your works list within the your Orchid sheet. And then for Zenodo, we use Zenodo for unmutable DOI generation of workflows, because there's the potential that, like on GitHub, you can change stuff and it can vary. Zenodo will take a snapshot of that and archive it there and make the DOI permanently accessible. Next slide, please. And some of the recent improvements, we have ongoing work on UI. We've done some improvements to our search and just general usability. We have improved our CWL support. We've done significant work on CWL 1.2 and continuing to add additional features for CWL. We have GitHub app support to make it easier to use GitHub with DocStore. I already mentioned the Orchid ID, and then also the DOI generation with Zenodo. In addition to that, some of the stuff we're focusing on for 2023 is workflow metrics. We're working with some of the analysis platforms to get statistics on how frequently or which workflows are being executed and how frequently they're being executed. We're looking to add support for registering Jupyter notebooks. That will probably have several different forms. We're still investigating that. We'll improve our local compute cluster, local compute and compute cluster support. And then in addition to that, we are working on improving our usability and accessibility of our documentation. The documentation has grown fairly organically over the life of DocStore, and we realize it needs some work. And we've got resources looking at restructuring it and making it more usable. So that's it for me, unless you have questions or comments.
All right, thanks, Ben. Do we have questions at all about DocStore? A lot of good information there. I know a lot of our researchers really enjoy having DocStore available to them for their workflows. Any questions? Okay, next up we have another researcher, Fei Wan.
Hi, thank you, Amber. Thank you. Oh, nice to see everybody, and nice to see some previous fellows, and it being well, and nice to see all the co-workers that we work on the platform here. And I will have highlight my research called Association Study of Iron Overload in Sickle Cell Disease Population use the TopMap data. So I work as a cohort 1 fellow on the platform, use the 7 Bridges platform. And I have some data very interesting developed from this platform. I use all the data directly on the platform. Actually, first I upload the data, but it's available in easy access now for the top med cohort there. Next slide, please. Okay. Oh, sorry. Maybe I'll show it all here. Yeah. So, and my work is, trying to find some biomarkers related to the called iron overload. Iron overload is a complication related to transfusion during sickle cell disease. When they have a stroke or they have some other complications, they have blood transfusion. But during time, many people, but not everyone, many people developed a complication called iron overload. And researchers have found some of the biomarkers related to this complication. And when we have access to the whole genomic sequencing, we want to see if there are more sensitive biomarkers can be found related to iron overload. We are thinking maybe it can be used later for physicians to use these biomarkers to treat the SCD patient with iron overload for early clinical identification and for treatment help. So on the right you see red top here is a Manhattan plot. It just want to see, okay, you see there in the middle it's saying chromosome 11. There's one big locus here is saying, okay, this is a chunk of biomarker or locus there is what we want to see. Now related to ferritin. Serum ferritin is a protein and have the iron there. We use it as an iron overload indicator here. Okay, so and when we lower the plot, just show you, okay, on this chromosome 11 locus, which genes are there, and so are they coordinated and inhibit together there. So on the red color means that they are herited together. So we are still in the process now trying to see, because sickle cell also have different genotypes. We want to see if the different genotype will be the biomarker in the different, because we have 4 cohort in my study. So we are still in the process now. Next click, Amber. Click not that. Okay, yeah. So we use this data that's use the Genesis platform that workflow on the SandBridge platform. So that's the we have that result. I have part of my abstract published on the blood. I think couple years ago, but I want to have more result to come out, and I will update later with our community here. Next slide, please. And so the components on the ecosystem I used is for the data is from the TopMed, the whole genomic sequencing, and from sickle cell disease, the cohort data there. So apps I use on the SandBridge platform for the BCF to merge all the VCF files, and I use a filter, and then I run a Genesis for the Manhattan plot, QQ plot. And I use the TopMed Annotation Explorer to see which genes are there, to see their function there. And I also use RStudio Data Cruncher to see, to do the phenotype harmonization, also to run the new model first. Okay, so and also a collaboration is I can add all my collaborators, from multiple institutions on my the projects on the platform. And Amber, next one, Amber. Thank you. And so I think which helped me for the for my research is I've been struggling, to be honest, when I don't have these resources available. We were thinking to buy some powerful machine, and also to look for some is there service on the campus to do this. Finally, we have this, we can access of data on the platform, and I have my data stored in the platform there. So, and I also found that when I run 4,000 genomic sequencing for the GWAS, it just takes more than half hour. so it have powerful computation power there. And like I say, I stored my data there, have the storage space there. And all the app there, so I think I use most of it, the app. It means that you can press a button, put your file, you can run. So they have the documentation there. And although some met the app is because not some app I use it as a first user, I have to to see, okay, maybe this part that we have to pay attention to make put more details. But mostly all the documentation you can follow on to run the apps. And even there, if some trouble comes up, you can press the help button to access to the help desk. And the other is I found is a very close connected research community on the platform. So I've been working with on the with Amber, Emily, and Dave, and also when I have trouble, usually I reach out to Dave. I think initially very much, and also the help desk. I think several colleagues there they helped me during the troubleshooting there, and we have the fellow, like we know we have the fellows, the quarterly meeting, and we have the Slack channel. So I feel we are, close connect research community here. Next slide, please. And advice is, I think at the beginning, when you jump to the platform, it's a little bit overwhelming because so many resources, and including data, apps, and something you know, something you don't know. Okay, but you can either from your own paper or from your own work plan, you have an idea, you just focus what you want to do. Then you found that all under help desk help, maybe, and you found that app or that step, you follow the instructions to use them, to try them. And at the beginning, I also tried to troubleshooting, several times by myself. Sometimes I can get it resolved, but if not, not hesitate to reach out to the help team. Okay, and always, I know sometimes you will feel frustrated because something won't work at the moment, but don't feel frustrated. It's normal for the data sets. I think for every research, it's the same thing, right? So, and write down notes. I mean, because after a while, is you don't know where you saved your file. That will be one thing, right? And also, you might you run something smoothly this time, it doesn't mean next time it will run smoothly. That means that you will just forget a little step or something you didn't pay attention to. So I write down the notes where I'm doing those things. It helps me later if I want to run another time. Okay. And the other advice will be engage with the research community. By doing this, one thing we can help each other. The other thing is reduce your stress level a little, but maybe sometimes increase. But I just think it's helpful to interact with people and build your skills, build your confidence. Yeah. So that's my advice here, and I will stay here if you have questions, and we can chat later. Thank you.
Thank you, Feiwan. Good advice as always.
Yeah.
Do we have any questions for Fei immediately? Again, feel free to unmute yourselves. Okay. So we have Seven Bridges update next. I think we had some slide updates while we were presenting. So I'm just going to do a quick refresh. I think we should be good. I'll go back into slideshow mode. Hopefully you can still see my screen. And we will, perfect. Okay, we'll let Dave go through the updates for Seven Bridges.
Thank you. My name is Dave Robertson. I am the community engagement manager for BDC powered by Seven Bridges. I want to thank Fei. She's a fantastic teacher, and she helped us give a workshop at Howard recently. And she's a great teacher, especially if you're working on GWAS. So since January 2022, we have a wall of text here about all the things that have been updated. I mentioned Study Variable Explorer in coming up, but we have an improved Annotation Explorer front end. We have interops with Anvil, so that's been a theme this year, interoperability. You can actually import T-text data into your 7 Bridges projects without actually having to move the data, so that's very handy. We have a new interactive web apps gallery, which I'll talk about. You can now link Azure Cloud buckets the same way you could also link AWS and Google. So that feature is called volume. So if you have a volume of data, you can link it directly to your 7 Bridges account without having to actually move the data anywhere. We also have WDL and Nextflow support, so you can run those in your project. But for those two, you have to request early access features. So let me know if you want to test those. And over the past year, we've added over 100 new tools and workflows. Next slide, please. Okay, so Study Variable Explorer. So this is a new feature. It's a completely graphical interface. And we have a guide that you can click to when you get the slides. So the purpose of Study Variable Explorer is to allow you to search and compare phenotypic variables just very quickly without having to log out of the platform. And a great thing about it is you can compare the variables, but you can also tag them and write little notes to yourself about how these phenotypic variables could be useful in your research. Next slide. So Annotation Explorer has been around for a little while. It's Faye mentioned it a little bit. It's very powerful. It allows you to browse. There's different data sets. One of them actually is the entire genome, so you can browse annotations for every position on the entire genome. There's also a top med freeze 8 and a freeze 5, and an open data set as well. We've improved the front end, so you can immediately start browsing and filtering these for these annotations. So for instance, the top med freeze 8 has 440 annotations that are coming from the typical genomic annotation workflows. And that ends up being 5.2 billion rows. We also have a genome-wide data set, which ends up being 52 billion. It's telling me 52 billion rows, and the genome-wide is 701 variant annotations. I put an asterisk by export. If you want to export files to use in a GWAS or any data frames from here, you still have to use the previous version in RStudio, but that's going to be corrected, I believe, upgraded in 2023. Next slide, please. So of course, you can launch RStudio, Jupyter Lab, or SAS as well. So that is still an interactive feature that you can use. Next slide. But if you are not so much wanting to do the coding and you want to explore some more graphical interfaces, we have an interactive apps gallery, which has 3 items so far. But if you are a developer and you want to host one of the apps, especially if it's in the R-Shiny language, please let us know. We can host it. So currently we have Genesis Model Explorer, Locus Zoom, Shiny app, and a new one called Omics Circos, which is based on a bioconductor package. What this allows you to do is make those great circular plots very easily without any coding required. Next slide, please. So we have currently over 700 tools and workflows in the public apps gallery. Genesis is very popular, but we also have apps to work with CRAM files, to do RNA sequencing, QC, and machine learning. And those are all available in a graphical interface. So that is my last one. Any questions?
Thanks, Dave. Again, we'll open it up for questions if you have any. I know we typically end at 2, but we have a few more updates. So people don't have other engagements to get to, we're going to keep going through. We have one more fellow coming up right after Dave. And if there's no questions, we will jump in. I'll pause for just a moment, maybe 5 seconds. If you have a question, unmute yourself. Okay, otherwise we'll jump right into Daniel's research. Thanks, Daniel, for joining us.
Yeah, great. So I was am a cohort 3 fellow, and my project is working on the same data set that you heard from Jasmine. And I'll be looking at the non-malaria side, so looking more at the human genetic diversity and trying to find genetic variants that impact cardiometabolic traits. So next slide, please. And so our data set is a TopMed project called the Africa 6K, and it has about 6,392 samples. And this is a really unique data set to be able to study human genetic diversity, because not only does it have a large number of samples, but it has a very large amount of ethnic diversity, with over 200 different ethnic groups coming from Botswana, Cameroon, Ethiopia, Guinea-Bissau, Ghana, Kenya, and Tanzania. The first part of my project is to work on characterizing this genetic diversity by using a program called Admixture, which can give us an idea of the genetic ancestry, although it's more of an approximation of the actual genetic ancestry, which is what this figure here is showing. This is from an older paper. I don't have a figure of that to show you just yet, but the idea is the different colors represent different admixture modeled genetic ancestries. And so the quantity in that pie chart would represent the amount of that admixture genetic ancestry found across different areas in sub-Saharan Africa. And we need to understand the genetic ancestry so that we can understand how we want to group different populations in our samples in our data set to be able to actually do other types of comparisons, which would be my next aim in my project, which is to run different scans of positive selection using a program called CellScan. And so this will help us identify variants that might be have more of a biological function if they're under positive selection. And the idea is that if we can look for a bunch of these different selection signals and run something like a gene ontology analysis, we might find different populations have different selection signals that might be related to cardiometabolic traits. And then the final element of my talk will be of my project will be to combine our genetic data with some proteomics, which we're using with Olink. So we'll have about 3,000 protein biomarkers, which will then be able to look at if their different levels are associated with these scans of positive selection, as well as other genetic variants in the TopMed Africa 6K. And then the final element that I forgot to mention, if we look at the top table, we've also combined the Africa 6K dataset with some of the publicly available samples in the TopMed Freeze 10 dataset, which has over 4,000 samples from the Human Genomes Project, Human Genome Diversity Panel, and the 1,000 Genomes Project to help us get a sense of how the populations in Africa compared to other populations from elsewhere around the world. Next slide, please. And so how I accomplished this project, all of my analyses were run on the 7 Bridges platform. And on 7 Bridges, there's a public admixture app. So I was able to use that with some slight modifications so that I can control the random seed function so that I can run a lot of different replicates of admixture for different numbers of ancestral populations. And then I also had to develop my own app to be able to run CellScan and all the different selection scans that CellScan can input. And I received a lot of help from Dave on that because I was struggling with a few of the elements since it was the first app that I ever composed for the platform. And so being able to get help from the 7 Bridges stuff was very helpful. And that's all I have to present.
Thanks, Daniel. Questions for Daniel right away?
I'll ask a quick one, I guess, if that's okay.
Yeah, of course.
Hi, this is Nathan Bowen at Clark Atlanta University. So I'm interested in local admin.
Oh. Sorry, I think I muted you on accident. I went to mute myself.
It's okay. Where did I stop?
Apologies.
I heard local, I think local admixture or adaptation. I'm not sure.
Yeah, no,
I heard local.
So ultimately I mentioned local admixture, but I'm curious on yours. Do you have to maintain storage for your reference populations, especially as you introduce the new ones from within Africa? And does that chew up a lot of your compute?
Yes.
Resources. Thanks.
Yes. Admixture is surprisingly very expensive to run. I've run it before, but not on a data set with 10,000 people. And so some of the larger K values are taking more than 6 days to run, so it becomes quite computationally intensive. So yeah, I guess I didn't have to do too much updating with storage, but it was something to be aware of because it does become quite computationally intense.
Yeah, thank you.
Markers to use. I see a question. So this was across the genome. So just chromosomes 1 through 22, but we did heavily filter to try and reduce the size. I think in the end, I kept any variant with a minor allele frequency of at least 5% or greater, and also heavily LD pruned the data to get the markers down to about 1 million. And even then, it still is quite computationally intense.
Good questions. Any other questions for Daniel? Okay. Moving on, we have some Terra highlights and updates. Unfortunately, our Terra rep, Elisa Manning, had to jump off to another meeting. So we'll go through her slides. I won't be able to do them justice, I'm sure, but she did list a few Terra highlights, fast facts. So since January 1st, they have 8 new featured workspaces, plus updates to the featured BDC workspace. You can see the numbers here on workflows launched with just over 1,000 on BDC. On the notebooks launched, again, a little over 1,000 on BDC, and data sets imported with 94 on BDC. And they reached a milestone of 1,400 average monthly users. Really good highlights. New features, smarter workflow launching, reduces latency and improves user experience, and now supports more analysis applications. So they currently support Nextflow and CWL, working towards supporting UCSC Genome Browser and the Hail Batch service. And there's details on their roadmap for the support on the slide. Again, sorry, I could not get those into the chat, but if you go on the forum, the links are there. And they have a new analysis tab, so an improved UI to make interactive analyses more intuitive with their analysis tab. The Notebooks tab has been replaced with this analyses tab, a clearer path to configuring and managing cloud environments, and again, details at the link on the PDF. Okay, well, I thank everybody who is still here. I'm going to launch one more poll. I promise it will not take long. If I can figure out how to do it. There we go. Okay. So I'm launching a quick poll about networking in 2023. Another thing we've been hearing is that researchers would like to be networking. We're just interested if that sounds interesting to you. And if it does, if you could let us know what that sounds like or what that looks like for you. What's the ideal networking setup? Do you want to be talking to people with similar research interests? Would you like more community hours based events where we plan and host them and you just join? So there is no wrong answer, of course, but let us know how we can engage you more and set up some additional initiatives for you in 2023. Again, we're a very community-centric endeavor, which I'll be talking about in a minute. I promise it'll only be a few minutes long, and then you guys can get to the rest of your afternoon. So if you could answer the poll, we'd really appreciate it. Building a community, we are growing. We have 485 community members that have joined the ecosystem. Just to look at the chart, you can see our request for credits, but also the blue line is our new community members. We had a spike in October, or going from the summer into October of 21, and we're steadily increasing. Ongoing endeavors that you are likely aware of is our monthly newsletter. If you do not receive our newsletter, it's probably because you haven't joined the community, so make sure to join. And we do have a YouTube channel that we are always adding more videos to, and we have a lot of big plans for this channel. In 2023, we have some reflections from fellows videos. So again, thank you to our 4 guest researchers today. We will be having videos from researchers just like them that were in the cohorts 1, 2, and 3, talking about their experiences with BDC. So if it was interesting to hear them speak today, if that was interesting for you, we will have more of those added to our YouTube channel next year. We'll also have all our community hours recordings there if you ever want to go back and visit something. As well as recordings from events and workshops, as well as how-tos and tutorials. We actually are planning a lot of educational materials that will be released next year, so that's very exciting. So make sure to subscribe. And of course, our community hours. I would like to thank all of our attendees, all of our co-hosts, and all of our volunteers that have made this possible. We've been having our community hours for about a year and a half now, maybe just under. Again, we post our materials on the forum, including recording slides and notes. There's a variety of sessions we've held on all sorts of topics. We revisit some popular topics so people can join and get the information live, but you can always go back and look at things on the forum. But again, just a huge thank you to everybody that participates. I see a lot of similar names each month, and it means a lot that you're attending. And I hope that you enjoy the information we present to you. And I want to thank all of our platform reps who make this possible and put in a ton of work to have really great slides and engaging material. None of this would be possible without you, and it would not be possible without our attendees. So thank you. Of course, if there are topics you want us to cover that we have not, all you have to do is reach out. I think most of you probably know my email. I will be putting it in the chat, or I will. But reach out to me, reach out to our platform reps, file a help desk ticket, post in the forum, whatever it is, and we will cover anything that you ask us to. So we're always looking for new ideas, and we want to know what you want to know. So just let us know that, and we can make sure it gets covered in one of our future sessions. So again, a huge thank you to everybody. Again, join the ecosystem if you have not already. And I know we're over time, but if you have questions, it looks like most of our researchers are still here. If you want to ask follow-up questions on their research, if you want to ask any questions about Biodata Catalyst in general, all of our platform reps are here. So now is the time. And of course, if you have to head out, thank you, and we'll see you next year. Watch your email for our next community hour sessions that will be happening in January. And I open the floor for any discussion, any questions, any comments.